Hello, welcome back. This is episode 8 of the Science Makes Sense series, brought to you by a Tutor's Voice Professional Tuition Service. I'm Jennifer, and I'm here to teach all you biologists out here today about that subject that teaches us about what makes you, and me, individual, superb, and unique. Yes, people, it's meiosis. So that's the process of cell division that results in the production of haploid gametes that, upon fertilization, result in the production of an embryo that looks like me, or looks like you, or looks like you, or you, or you, or you, or you, or you, or you. So, as you'll remember, in episode 6, we looked at mitosis, which is the process of cell division where we produce daughter cells that are genetically identical to the parent cell. If you haven't seen that video, pause here, have a look back at episode 6, because that will be really useful in terms of some of the stuff we'll discuss today. So you can see some similarities and differences in the different stages of these two forms of cell division. Okay, so let's begin. In meiosis, what's really important and unique to it compared to mitosis is the fact that you have a cell division process that involves only one replication stage and yet produces 4 cells. And those 4 cells are not genetically identical to the parent cell because they're actually haploid gametes. Whereas with mitosis, you produce daughter cells and you produce 2 of them per one parent cell, and they are genetically identical to that parent cell. So in meiosis, everything starts off just as mitosis does, in that you have the chromatin condensed to form chromosomes in prophase 1. But what happens in prophase 1 as well, in addition to that, is that the homologous pairs of chromosomes will align together to form a tetrad of these 4 sister chromatids. And then the neighboring ones will cross over to form what are called chiasmata, and a singular one is called a chiasma. And these chiasmata are extremely important in producing the variations that we see in individuals every day, in that some of the maternal aspects will then be shared with the paternal chromosome and vice versa. And that particularly you'll see in subsequent stages. So in metaphase 1, these homologous pairs that have been crossed over will now align at the equator of the cell, and the centrioles will move towards the opposite poles of the cell and will then project out spindle fibers that will join to the centromeres of the chromosomes in these homologous pairs. What happens next is anaphase 1, where the spindle fibers contract, pulling apart these homologous pairs so that each individual chromosome from that pair move to opposite poles of the cell. Then in telophase 1, what you start to see happening is that the membrane of the cell will start to pinch inwards until cytokinesis is complete, so that now you have 2 separated cells where you have a mixture of paternal and maternal chromosomes in each, but then you've also got these recombinant sections in those chromosomes as well. And so then meiosis 2 is very similar to mitosis in terms of what actually happens at each stage, except that the appearance of the DNA is obviously very different, and we'll examine why exactly that is, and obviously what types of cells we end up with. So during prophase 2, the chromosomes are condensed, and then in metaphase 2, those chromosomes will then align in the equator of the cell, and the centrioles will then move once again to the opposite poles of the cell and project out spindle fibers to join to the centromeres of the chromosomes. So that during anaphase 2, as the spindle fibers contract, the individual chromatids of each chromosome are then pulled apart and moved to opposite poles of the cell. And then in telophase 2, the cell membrane again pinches inwards and gradually forms 2 cells per one previous cell that we had from meiosis 1. So now we end up with 4 individual cells, each with sets of chromatids that have come from both the maternal source of chromosomes and the paternal source of chromosomes. And so because of the fact that the assortment of these chromatids in these individual cells and the fact that chiasmata formed during prophase 1, you now have 4 haploid gametes, each completely individual in their genetic makeup, and therefore making the wonderful individuals that we see in everyday life, including you and you and me. And so that's the end of the video. I hope this has been really helpful to you. Remember to leave your video requests in the comments box, as this channel is entirely for your benefit. I make these videos to ensure that every child everywhere is able to get the best possible results in their exams and have the best possible understanding of their subjects so they can love it as much as I do. Remember to like and subscribe to the channel because I make new videos every single week. Remember to follow me on Twitter, follow me on Instagram, because I share really helpful study tips to aid your revision in addition to sharing these videos. I will also be doing a giveaway really soon, so make sure you look out for it on social media. And please, 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 as always, share your Nando's with me. I'm starving hungry. It has taken a very long time to make this video. Let's all go and have fun and enjoy life. And I'll see you in the next video. Bye. This is episode 8 of the Science Makes Sense series. That's 2 thumbs. That's what I want. No, that's 9. I can't count, guys, but I can get you through your A-levels. Wow, that was it.